Dear students, Assalamu alaikum. Hope you will be fine. As in the previous video we have discussed about the transporting structures of the plant as xylem and phloem. Today we will study the internal structure of dicotyledonous stem. As you have already studied in the previous chapter about the internal structure of leaf. So here we are going to study the internal structure of dicotyledonous stem. So first of all, I will tell you what is mean by dicotyledonous. Remember one thing, that we have two types of plants. Some are called monocot or monocotyledonous, or some are called dicot or dicotyledonous. So what is mean by mono mean one, di mean two. So you have observed some of the seeds in the plants which are consisting of just one cotyledon. What is basically the cotyledon? Cotyledon is the reserve food material for the growth of a new plant. Okay, so you have observed corn seed. Okay, so you have observed corn seed. It is consisting of just one single cotyledon, so it is called monocot. In the same way you have observed rice, it is also monocot. You have observed wheat, it is also consisting of one single cotyledon. And in the same way you have observed dicot, di mean in which two cotyledons are present. For example, you have seen chickpea. Okay, so chickpea, it is consisting of two cotyledon. Okay, in the same way you have seen peanuts, you have seen walnut, you have observed many such seeds which is consisting of even almond. They all consisting of two cotyledons. Okay, so those which are having two cotyledons are called dicot or dicotyledonous, and those who are having one cotyledon are called monocot and monocotyledonous. So there is some structural difference in them. Their arrangement of vascular bundle is different. For example, the leaf of monocots, they are always long and narrow, and the vascular bundles are always present just here. These are the vascular bundle in this form. And in case of dicotyledonous, the leaf is always broad and it has a netted or network of veins is present. So this type of venation is called reticulate venation. Okay, so in this way there are also many other structural differences, so we will discuss when we will study about them. So here I am going to explain you the difference between not the difference, basically the structure of dicotyledonous stem and dicotyledonous root. And you will remember the arrangement of tissues in stem and root, and also you have to keep in mind the arrangement of all these structures in a leaf as well, because their arrangement is different. The arrangement of vascular bundle is different in leaf, it is different in stem, and it is different in root. As you have seen in the structure of stem, here you can see, you know that upper epidermis is present, then the lower epidermis is present. I am not making in detail, just to make it clear. Okay, and you already know about these tissues. Spongy and palisade mesophyll tissues are present. So here I basically want to tell you the arrangement of vascular bundle. Here in case of leaf, you have studied that xylem was present in the upper side and phloem was present here on the lower side. This one upper was xylem and this lower was phloem. Both are making vascular bundle. But now when we'll do study about the arrangement of stem, so here this arrangement is different. For example, this one is a stem, and if we take its transverse section like this, and we will draw here a transfer section like this. So here I'll tell you the arrangement of all different parts. So as in case of here leaf, you have seen upper epidermis and lower epidermis. So here epidermis is present. The outermost layer is the epidermis. Okay, I'm not making the all cells, but just I'm showing some region of this stem. Okay, this one is epidermis. Then the next layer is the tissues. Few layers of these tissues are present. Remember one thing, in epidermis no intercellular spaces, but here in this layer there are intercellular spaces are present a little bit. So this loosely arranged cells are present. So this layer is called cortex. Okay, then a layer is present which is called endodermis. The whole this layer is present here in this area. So this one is called endodermis. Okay, now here vascular bundle is present in ring form in the dicotyledonous stem like this. Okay, so here I'll show you that and what is the difference here, difference from leaf to stem. Here in this area the phloem is present in the outer side region and xylem is present towards the inner side. Okay, this one is phloem, this one is xylem, and between xylem and phloem is here a layer is present and which is called cambium. This one is called cambium. Cambium is consisting of actively dividing cells which make new phloem and xylem every year. Okay, so at present just keep remember, phloem is present outer side, xylem is present inner side. Between these two cambium is present. Okay, so this one is the vascular bundle. Okay, here in the central region cells are present which are called pith. Okay, so you have to keep remember this arrangement. Okay, so outermost layer epidermis, then inside epidermis cortex is present, then the layer of endodermis is present, endodermis is present, then vascular bundles are present in the form of a ring, and here the phloem is present outer side and xylem is present inner side. Between these two cambium is present. Okay, now here you have seen in the leaf xylem was outer side, phloem was inner side, but here in the stem phloem was outer side, xylem was inner side. Okay, now I am going to tell you the internal structure of root, and in the root this arrangement is totally different. Okay, again we will take root, we'll cut its transfer section, and in this transfer section, so remember again I am making this as a root. Okay, but this arrangement will be again here arrangement you have to keep remember, it will be again different but nearly the same tissue. So what is going to happen here, the outermost layer is called epidermis. So remember one thing, here is a difference, a little bit difference. The difference is here you can see in the root hair cells are also present. So here these epidermal cells, they have outgrowth. This outgrowth is basically the root hair cells. And remember one thing, you will not make root hair cell like this and this. It is wrong. It is the extension of this cell like this. Okay, so here root hair cells are present in the epidermal layer, or you can say modified epidermal cells are present, and this layer is called, instead of saying epidermis, you can say even epidermis, along with these root hair cell it is called foliferous layer. Okay, again the internal structure is consisting of cortex cells. So here cortex cells are present. Okay, then here another layer of endodermis cells is also present. Okay, I'm not making the all tissues just to save the time. Okay, now remember one more thing, but here the arrangement of xylem and phloem is in the form of star shape. Okay, so here I can show you like this. Here you can see star shaped xylem is present like this. These here the xylem tissues, they are present in the form of this star shape, or you can see 5 arms of xylem is present, and between these arms of xylem here phloem is located. Okay, so here the phloem tissues are present between the arms of xylem. This star shaped xylem is present, and between the arms of xylem phloem is present. This one is xylem, star shaped xylem, and this one is phloem. So here you can find the arrangement is totally different. Nearly the structural cells, they are the same. For example, epidermal layer, then cortex cells, endodermis, then xylem and phloem, but the arrangement star shaped xylem is present, and in the arms of this xylem phloem is located. Okay, so remember one more thing, in case of stem, outside the epidermal layer again cuticle is present to prevent water loss as it was present in the leaf. Okay, but in the root there is no cuticle. Why? Because in stem and in the leaf there is cuticle to prevent water loss, but root is present inside the soil, so there is no need of prevention of water loss. So here you can observe the structure of xylem and phloem as they have shown you. Here this is the structure of stem. So here you can see in dicotyledonous stem the vascular bundle is present in ring form, and phloem is outer side, xylem in inner side. In between here cambium is present. Okay, but in case of root you can see root filiferous layer is present. Why filiferous? Filiferous layer is basically epidermis along with the this extension which is the root hair. Here you can see there are some root hair cells which are they haven't draw correctly, they are wrong, but whenever you have to draw you have to keep remember that this root hair cell is the extension of epidermal cells. Okay, so now you will also try to draw these and even draw in your notebooks because these structures, their location, their position of xylem and phloem is very important from Cambridge paper point of view, and if there is any problem you can ask. Okay, Allah Hafiz.